In this procedure, a new near-infrared fluorescence catheter, or NIRF catheter, is used to image two-dimensional intra-arterial vascular biology and monitor biological processes including atherosclerosis inflammation and angiogenesis. First, an experimental model of atherosclerosis in the aorta and iliac of a New Zealand white rabbit is generated. Next, at the appropriate timing, an NIRF activatable nanosensor is injected to label an inflamed plaque. 24 hours later, a co-registered arterial angiography, intravascular ultrasound, and in vivo and ex vivo intravascular near-infrared fluorescence images of rabbit atheroma are acquired. The aorta and iliac arteries are then dissected out to perform ex vivo fluorescence reflectance imaging immunohistochemical analysis of the inflamed plaque. The final results show quantifiable signals that correlate to an inflammatory atherosclerotic plaque.
This method can help answer key questions in the field of molecular imaging, such as in vivo real-time detection of inflammation in plaques, key markers of vulnerable plaques, or the precursors of acute myocardial infarction. The implications of this technique extend towards therapy of atherosclerosis because it can allow real-time in vivo detection of inflammation, which can translate into target-specific and patient-specific therapies that reduce inflammation in human coronary atherosclerosis, a key to preventing the dreaded acute myocardial infarction. Though this method can provide insight into coronary atherosclerosis, it can also be applied to other systems including peripheral vascular disease, for example, to characterize lesions in the carotid, aorta, iliac, or femoral arteries, the culprits of peripheral vascular disease.